Thank you. So my name's Grace Pendlebury and I work on the Australian genomics platform called Shariant. And today I'm going to be presenting some recent survey findings from work we did within Australian and New Zealand diagnostic labs on their somatic testing and curation practices. So I work from Brisbane, which is in the southeast of sunny Queensland in Australia, and while the survey work that I'm presenting on today was done nationally, specific to these labs, the topics translate internationally and provide some insights and barriers into the state of implementation for somatic testing labs. So to provide some context, while Australia is almost the size of the US in landmass, our population's actually less than that of the whole state of California. So not only is the volume of testing much smaller, but the Australian and New Zealand healthcare and pathology services also operate on a model much closer to that of the UK and Canada than the US. So in Australia, while some patients do pay out of pocket for expedited private testing or anything that's not included in the Medicare benefit schedule, the majority of the testing is publicly funded through that federally managed Medicare system or distributed by individual states and territories through their public hospital funding.
Can you just move your mic a bit, please?
Yeah, sorry. Is that better? Okay. So for those of you who might not be familiar with Shariant, it's a web-based platform conceptualised and designed by my colleagues to automate the sharing of clinical variant interpretations and evidence codes between diagnostic labs in Australia and New Zealand, and it's a closed access system. So at the moment, we've got just over 31,000 germline variant submissions shared across 13 labs in the system. And we also detect and notify labs if their classification is discordant to that of another and provide a platform for them to hopefully resolve that discrepancy. We also connect to the ClinVar API on behalf of the labs to submit their variants, and that's actually driven a 2,000% increase in Australian diagnostic ClinVar entries since 2009, most of which came directly through Shariant. So we're pretty proud of that. And if anyone wants to know more about the platform in general or visit our GitHub, I've included the PubMed ID of the paper on the screen that my colleague Emma Tudini published in 2022. So until recently, Shariant has only supported germline variants, but we've just completed an expansion project to offer support for somatic. And this started with a landscape survey of curation guidelines and of the labs doing somatic testing in the region. And we've covered a range of areas in this, but what we wanted to find out was firstly the scope of their services to understand the context of testing being done and what evidence fields we might need to build into Shariant to support that, as well as anticipate the kind of volume of records that we'd have to handle from a computational perspective. Now, in the germline space, the ACMG guidelines, as you know, have been fairly well ingrained for some time, and we were anticipating that this would be a lot more heterogeneous with somatic labs. So we wanted an understanding of what types of curation schema Shariant would have to compute and compare between if we onboarded them. And we also wanted to know what the current sharing practices already were for somatic labs submitting their curations to any knowledge bases like ClinVar to assess the gap that we're actually trying to fill regionally. And then to make the platform as useful as possible, which is always the goal, we included some questions for labs on their preferences for features. So we used the national accreditation bodies to compile a list of 40 labs to contact, and we were looking for a minimum of gene level accreditation. So we were hoping to catch NGS labs, and of those 40 that we contacted, 27 groups ended up participating in the survey, which was just about a 70% response rate, which we're really happy with. And you can see on the right map there that we had quite an even distribution across regions as well. And then to do the analysis, we grouped labs into categories according to the types of services they offered. So the vast majority were that mixed discipline cancer service labs, but we did also have some heme specialists and labs doing non-cancer somatic work, mostly for vascular malformations and regional overgrowth syndromes. In terms of testing scope, most somatic NGS was being done with multi-gene panels, So only 2 of the labs that we surveyed were offering accredited whole exome sequencing, and none were offering accredited whole genome sequencing. So you can see in orange on the left there, there's actually only 1 whole genome panel that was being run, and that was a non-accredited research use only. We also asked about the types of variants that were being captured. So out of 4 categories: single nucleotide variants, small indels, copy number variants, and gene fusions. We found that only 36% of the labs were actually capturing all 4 of those variant types. And in dark blue on the right, you can see that just over a quarter, 28% of labs were only capturing single nucleotide variants and small indels, which is particularly interesting given that, as you know, in oncology, there are a number of copy number variants or gene fusions that do have indicated matched therapies. So for the cancer labs in particular, when it comes to their curation pipelines, not unsurprisingly, we did find that it was mostly manual for 88% of labs. But most of them were performing curation for both the oncogenicity and the clinical significance of their variants, which was great to see. And then at the time of survey, it had just reached about 50% of the labs using the ClinGen CGC-Vic oncogenicity SOP published in 2022. But a number of the labs who were using different guidelines at that time told us that they were about to change or were evaluating the guidelines with the intent to change. So we've included a chart on the right there about the projected increase to a 70% majority that we're expecting over the next year or so. Looking at clinical significance across all of the disciplines, that was a much clearer majority, with 74% of the labs who perform this type of curation using the AMP-ASCO CAP guidelines. And if you split that back down to cancer labs, it goes up to 94%. So on the inner ring of this chart is the base guideline that they were using, and the outer ring indicates if that was in an unmodified fashion or if they were supplementing it or making any in-house modifications. And I will note as well that the labs using the ACMG 2015 guidelines in dark blue for clinical significance curation were the 2 who told us they were only performing somatic testing in complement to their germline service and in the context of pathogenicity. So it's not inappropriate for them. We also surveyed on reporting of clinically relevant biomarkers, and we found that less than half of cancer labs were reporting on things like tumour mutational burden and microsatellite instability. One caveat here is that given reporting practices were not a focus of the survey, without more information on individual assay accreditation or capabilities, we can't say whether the labs not reporting on any are doing so out of choice or because they don't actually run a panel large enough to validate the calculations. But it does highlight disparate reporting practices between labs nonetheless, in an area that could benefit from best practice guidelines. For therapy matching, 65% of cancer labs were reporting funded therapy options listed on the MBS, so that's government funded therapy options, but that's also still 35% who are not, though those are likely to be standard of care therapies anyway, which the referrers should be aware of. Under 50% of the cancer labs, though, were reporting on privately funded but still on-label therapy options and national clinical trials, which went down again to 24% for international trials. And then that leaves about just under 30% of cancer labs who responded none to this question, meaning that they aren't listing any therapy options in their clinical report, which I think is also an area for standardisation. A final striking, but not, again, sadly unanticipated result is that only one of the labs that we surveyed is actively contributing any of their variant interpretations to an international knowledge base such as ClinVar or Civic. And even that one lab, it was only if they had time for rare variants, and we all know nobody does have time when it comes down to the manual process. So this is the gap that Shariant can help fill, and we want to provide the same service for somatic labs that our germline groups are already seeing the benefit of. So we designed the survey originally to inform the somatic development, but what's actually come out of it is a pretty unique snapshot of the Australasian somatic landscape at this point in time. And what I've presented today is only a small portion of what we actually covered in the survey, and there's still more to investigate. such as the relationship between panel accreditation and reporting practices and the improvement of somatic variant sharing. Variant curation, as you've heard today, is still very much a manually intensive undertaking and can be a bottleneck for many labs. We hope that the learnings from this can inform national and international priority areas for standardisation. And this could include things like somatic genomic implementation policy as larger oncology panels are being adopted, and how these should fit within existing healthcare structures, particularly public ones like Australia and New Zealand. It also supports the development of somatic clinical reporting standards to establish global best practice for things like inclusion of biomarkers and therapy options in testing reports, with the ultimate goal of all of that being equitable access between regions to precision medicine and the improvement of patient care. As always, there are many wonderful individuals to acknowledge who supported this project. First and foremost, my colleagues Emma Tudini and Mandy Spurtle. Shariant is also a multi-institute collaboration between Australian Genomics at the Murdoch Children's Research Institute, QIMR Berghofer and SA Pathology. And we always have the biggest shout out as well to our fantastic user group of labs and all of the labs who participated in the survey. Thank you.